Where's my allergy pills? All right, it's allergy season, everybody. Woohoo! And it's time for TWIS. Nothing to sneeze at, just a science podcast that's coming up right now. And if you have not yet subscribed to things, make sure you hit all that subscribe and like, and make sure you get all the notifications right now, because you want to get this every single week. This show right now is going to be the live show. We're going to edit it down a bit afterwards, and that'll go out as the podcast. So you are getting the full, unedited, warts and all version. Starting, I don't know, Justin, starting about now?
Yeah. That's the moment in which you can do.
Starting in 3, 2, this is TWIS. This Week in Science, episode number 829, recorded on Wednesday, June 16th, 2021. Why are bats disease vectors? Hey everyone, I'm Dr. Kiki, and tonight on the show, we are going to fill your heads with filaments, microbes, and bats. But first...
Disclaimer. Jon Stewart recently went on the Late Show with Stephen Colbert and suggested COVID-19 had escaped from a lab in Wuhan, China. And that might be true. It's possible for that to have happened. Things like that have happened before, because even a level 4 biosafety lab is only as safe as its weakest followed protocol. Also, it happens to be located in a country without a free press, where the government controls information like an authoritarian cheerleader mom with next-level deepfake skills. Also, it might not have. The Spanish flu of 1918 most likely started on a farm in Kansas. It just got identified in Spain. But John isn't anti-farm. In fact, he has one. One of the reasons there are often emerging novel coronaviruses from East Asia is not because East Asia has an inordinate amount of labs. It's because that's where 1 in 5 humans live. Greater numbers of humans, denser urban areas, leads to higher number of novel pathogens, making the leap into humans. Add in a temperate environment with some help from global warming and other things science is trying to save you from, and you have near constant conditions conducive to transmission. There are many thousands of coronaviruses in bat reservoirs around the world. Globally, bats represent 1 out of 5 mammal species and 40% of the mammals in East Asia. These viruses don't affect the bats, but they can be lethal to humans in extremely rare cases, which is why studying them is so important. And yes, there is a lab that studies them in Wuhan, a city with 2.5 million more people than New York City. And yeah, maybe they should put a lab like that out in the country, near, say, a farm in rural New Jersey, to keep them better isolated from human civilization. But what's worse, John blamed science for COVID-19, saying the pandemic was more than likely caused by science, which, stop. Anti-science talk like that is hurting America. It's why we can't get meaningful action on global warming, it's why we relied on Russian rockets to go to space for a decade, and it's why the pandemic hit the United States so hard, and why it still isn't done spreading. So stop. Stop hurting America. And think of what halting all research on viruses would even look like. We knocked out smallpox, chickenpox, measles, mumps, rubella, and polio. Remember polio? No, because you didn't have to live through it. But the people who did were losing their children to these diseases. So they fought back with science. What about AIDS? Should we end all AIDS research? It's a race for the cure, not a run from the research. And yes, working with pathogenic viruses is very dangerous. The frontline researchers who study them know this all too well. There's a constant risk that a pathogen will escape containment despite the multiple layers of precautions that are in place. And they occasionally fall ill from the very thing they are studying. It's dangerous work that scientists do while responding to these threats. But these are people who care about the experiment of humanity and keeping it viable. And the next time you want to blame science for something, look no further than This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science?
to you, Kiki, and hey, where's Blair?
Oh, Blair's out for tonight. She's not feeling well, so I hope you all send her some well wishes. But we are here with good science for you at This Week in Science. I hope you're ready for another amazing episode, well, full of science, because that's what we're going to talk about. All sorts of scientific stories. I have whirling filaments, plastic microbes, and I've also, at the end of the show, if we get there, I've got zebrafinch songs. What did you think, Justin?
I've got misleading research titles, Why Hoppy Beer is Good for You, Swiss Bat Viruses, and Underwater Archaeology.
Fantastic. We're also going to be joined by not one, not two, but three amazing researchers who are here to talk with us about the immune systems of bats and what makes them so amazing. And we will be speaking with them in just a little bit, so stay tuned. And as we jump in the show, I just want to remind everyone that if you have not yet subscribed to This Week in Science, you can find us all places podcasts are found: Spotify, Pandora, Stitcher, Spreaker, TuneIn, all the places. Look for This Week in Science. You can also find us on YouTube, Facebook, and Twitch. We're also on Instagram. And our website is twis.org. Okay, Justin, are you ready for science? Yeah? Okay, let's start with whirling dervishes in space.
What? Sufis in space? How did they get up there?
Yeah, no. Whirling structures in space. This is such a fun discovery. When you think of swirling and whirling objects in space, what do you think of?
Oh, well, I just think of slow motion, but it looks like it's spinning. It's like a galaxy.
Right. Galaxies, they've known very slow spin to them.
I don't know how fast they are moving. They might be moving really fast.
Well, it's fast, but not that fast. I mean, on a galactic scale, yeah. And then we've got black holes can have some spin. Pulsars have some spin. Like, there are objects in space that have spin, but when the Big Bang occurred, spin was not really this thing that was imparted on anything. Like, angular momentum was not that. It was a big cloud of dust, and then the dust started congealing into things, and in that process, all sorts of structures came out of it, and some of them bumped into each other and started spinning because of that. And in the structure of the universe, there are also galactic filaments. These galactic filaments, they stretch between areas of space, and they contain galaxies. They contain dust and gas, and everybody just kind of always went, yeah, they're filaments. They're not going to be spinning. But hey, look what you see when you actually go looking for it. Researchers just publishing in Nature Astronomy have said that they have possible observational evidence for cosmic filament spin. And so these massive filaments that contain galaxies, they're massive, are spinning at over 200,000 kilometers per hour. Like, they're spinning. Large scale
For those not familiar with kilometers, 200,000 kilometers is approximately the same as 200 million meters. So that's really moving.
It's moving, right? These are large objects in space, moving very quickly. And these researchers have said, hey, we never really thought this was a thing that spun, because why would it? Why would these filaments in space actually spin? And now that they know that they do, they have discovered that, yeah, there's these big spinning filaments in space. And so the big question is why? And they don't know why. And so that is now going to be one of the big questions that they will be looking for. They think that there are powerful gravitational fields of the filaments as they pull on gas and dust and other material, and that creates shearing forces that result in spin, but they really have no idea. And they're going to be looking for it.
Now that you mention it, I can't think of anything in space that isn't spinning. Solar systems are spinning, planets are spinning, even down atoms even have spin. Electrons have spin. Everything's spinning. Wow.
Why is it spinning? My mind is spinning with the possibilities. Tell me about some mice.
Yeah, this is just my quick story for the night. These researchers study published in Public Library of Science Online Biology by Marcia Trunifol at the Humane Society International. They were looking at titles of stories that get picked up by popular press. And they found that if in the title you don't say mice, if you don't mention that it's a mice model, it's tremendously more likely to get picked up and run with as if it applies to humans immediately. Meaning most journalism is not reading beyond the title of the paper. Yeah, so this happens apparently a lot with Alzheimer's. Mice don't get Alzheimer's, but we have mice that can exhibit Alzheimer's-like qualities, and we can do research on them. And one of the things that's also true about that research is that almost none of the studies that are done in mice translate into humans under this modelage. There's things that are learned along the way, but it's not that it will have that effect on humans. So, yeah. So you have to know whether or not, because it's true, at some point we're going to cure probably every disease known to mouse kind. And even some diseases that they've never been able to get, we will cure in mice. But it doesn't always translate into humans. However, in doing so, we learn pieces of the puzzle that we can later put together on the human model at some later date. But yeah, just leaving mice out of that title makes people think that it applies to humans immediately.
So this is starting from the research itself, though. If the research doesn't have mice in the title, then journalists...
In the title of the paper.
Then journalists don't report it correctly, and then people reading further. It just is a bad game of telephone all down the line because people forgot to specify. But that's just...
I guess.
That makes me sad for the field of science journalism and makes me actually
I don't even know if it's science journalism. I wouldn't even put it there because it's not like these are... It's pop culture journalism. It's not even journalism. It's just
so they're not really journalists who are trained to talk about science.
No, but they will appear in a local news segment, which you assume has journalists behind the scenes. So this is just part of the problem. So it's just kind of just encouraging maybe researchers to put mouse model in mice.
Yeah, at the end of every paper title about what they've done with mice, ... in mice.
Yes.
We cured cancer, ... in mice.
In mice, yes.
Yeah. Oh, my goodness. Oh, reporters, do your work. Make it better. It's sad that the scientists have to clarify that in the title.
Next story I got is out of Oregon State University. Researchers found a pair of compounds originating from hops that can help prevent the buildup of fat in the liver, which is huge because apparently there's like a quarter of people in the world that have this fatty liver disease. Often it's brought about by drinking, but people who don't drink can get it as well. There's a non-alcoholic version. That's 25% is made up of both of that. But the good news is hoppy beer prevents liver failure, and it's now safe to overindulge.
No?
Yeah. In the study, 60 mice were... Oh, man! It's a mouse study!
There you go.
All right, well, in a mouse model study, researchers showed that the compounds xanthohumol and tetrahydroxanthohumol, abbreviated XN and TXN, can mitigate diet-induced accumulation of fat in the liver. So they gave these mouse models a low-fat diet, a high-fat diet. They supplemented some with the XN, a high-fat diet with the XN, high-fat diet supplemented with even more, and then a high-fat diet supplemented with the TXN. And they got really positive results. So this is the beginning of, as we often talk about, anything that's sort of medical and when you're getting that first exposure to some research. This is the first step in a mouse model to finding a possible pathway for, in the right conditions, a future drug or treatment for fatty liver. But it was some pretty positive results in mice.
In mice. Yeah, there's so often things don't work in humans when they have worked in mice, and there are many reasons for that. We don't know all the reasons.
We don't know all the
for
reasons.
that.
One of the simple ones is, especially if you're talking about a compound, could be how our microflores and metabolisms utilize or take up or distribute these compounds can be different in an animal model and a human model. I mean, there's lots and lots and lots of factors. However, sometimes it just works.
Yeah. Yeah. So you're putting your hopes on this being one of the one of the pieces of mouse research that makes its way very easily into humans.
At this point, I'm still picturing this future where the Doogie Howser mice have inherited the Earth from the remnants of humanity, and they've gone back and they've read all of our research, and they now it's just one more thing mice of the future won't have to worry about.
Just one more thing. That's all. Just one more. Not in mice, but in chemistry, researchers have been working so hard to get rid of plastic, right? How do we get rid of all the plastics that we create? It's a real problem for our environment. Well, one of the kinds of plastics that is very prevalent is polyethylene plastic, PET plastics, or polyethylene terephthalate, more specifically, is one of the types of plastic that is very abundant and is used around the world. And researchers have been trying to figure out how we can possibly use microbes to digest the plastic, turn it into something else useful for society, and thus save the world from being drowned in our own plastic garbage piles. Reporting in Green Chemistry, researchers Joanna Sadler and Stephen Wallace have published their paper, Microbial Synthesis of Vanillin from Waste Polyethylene Terephthalate. So what they've done is they have genetically modified E. coli, which is a very common bacteria that's used in laboratories around the world, and they've created a novel pathway within E. coli. They shoved a whole bunch of instructions into this bacteria that aren't normally in E. coli, and they said it was very complicated to actually get all of the steps in place for this novel chemical metabolism pathway within this bacterium, but they did it. And they have, in sense, they have a single pot. They showed proof of concept from a plastic bottle that they picked up off the ground in wherever they are in Edinburgh, and they shoved it in the pot where they had the bacteria. They have different processes to take off the products that kill the E. coli, that the E. coli don't like, but it results in the transformation of PET plastic into vanillin in a single pot. And vanillin, does this compound sound familiar to you, Justin?
Can I use it in a root beer float?
It's not the root beer float, no, but it's the flavoring of the ice cream that goes into a root beer float. So we can get vanillin, the compound that creates the odor of vanilla, from vanilla beans, but there is also synthetic vanilla. And synthetic vanilla can be used in food products. It can also be used in household products like candles and room sprays. It can be used in cosmetics. And the researchers think that since this is coming from this plastic waste and the process they're using, they don't know if they'll be able to make sure that it's clean enough to be food grade vanillin.
Well, you know where we used to get that flavoring?
I don't think I want to know.
Oh, yeah. I can't get fact checked because there's nobody else here. But I believe it
There's
came
only our chat room for fact
from the
checking.
anal glands of beavers.
No, thank you.
Yeah, so I think that
Anyway.
it came from bacteria on a plastic. Way better source. Yeah,
Way better source. Oh my goodness. Yes. So this is a very promising new bacterial, microbially enhanced process for reducing the amount of plastics wastes that we have that will result in a beneficial compound that can be used that is also in a lot of demand. People want vanillin. So this could solve 2 problems at once, right? And then my final story for our opening. Did I have another study for the opening? I'm not sure that I do here.
I do.
You do?
Yeah.
Yes. Okay, go for it.
Yeah, this is where to go. An analysis of 18 species of bats living in Switzerland has discovered 39 different families of virus, including some with potential of jumping to other animals, including, of course, humans. This is Isabel Hardmeyer of the University of Zurich, Switzerland, and colleagues presenting their findings in the open access journal PLOS ONE. It is exceedingly rare for disease-causing viruses to make the switch from bats to humans, so some viruses carried by bats may jump to other animals, which then undergo changes that allow them to be transmitted to humans. So monitoring of viruses harbored by bats around the world improves our understanding and our detection of those that pose a risk to humans. So they did a lot of genetic analysis, and they're proposing that this is a way you could just do it. You could sample bats, you could have the library that you track over time to see if anything is mutating towards a known previous pathogen. One of the things they did find was a MERS-related virus. So the MERS is the pandemic that we had back in 2012. And if you don't recall that epidemic, it's because it was much more lethal than the current one. So it had a harder time spreading because it kept killing the hosts. Now, the thing that they've identified that's related isn't going to do that. It would have to have some pretty big changes. But it's still enough to know that we have reservoirs across the world. And as I was talking about in the disclaimer, I didn't even know this. 20% of species on the planet are a bat, or at least of mammalian species are bats. That's crazy. I didn't realize that bats were that prolific. I had no idea. So lots of bats. They have viruses. They don't get sick from them, so we need to monitor them. And before anybody, we're going to be talking to bat people here in a minute, so I probably shouldn't say too much. But before anyone's like, we should get rid of the bats, that's not the solution at all. Because if we get rid of bats, what we get instead is
insects and other
mosquitoes.
Yeah,
we get
other vectors of death.
Unbridled, there's a cloud of mosquitoes who are also disease vectors, but are much more annoying.
Yeah, I find this study interesting because it's out of Switzerland, right? So you've got bats from different locations, and I think we always talk about bats that are in Southeast Asia. We talk about bats in China. We talk about that as being the hot spot, the hot zone, right? But there are bats everywhere, and they live in these huge colonies all around the world. And so I think that is another point of, we've got these genes and we've got these various diseases. And interesting question to ask our next guests as we talk about how do we monitor and what do we know and what do we need to learn so we can protect ourselves in the future. This is This Week in Science, and I would love to introduce our guests for the evening. We are joined tonight by 3 wonderful scientists. I would like to introduce Doctors Angelique Corthals, Liliana Davalos, and Diana Moreno Santillán. We're going to be speaking about bat immunity. Dr. Corthals is a biomedical researcher and biological forensic anthropologist at CUNY's John Jay College of Criminal Justice. Dr. Davalos is professor of conservation biology at Stony Brook University's Department of Ecology and Evolution, and Dr. Moreno Santillán is a postdoctoral researcher at Texas Tech University. Welcome, and thank you so much for joining us tonight.
Thank you for having us.
Thank you for having us.
Thank you for the invitation. Yeah.
I'm just really glad, like those segments that you had earlier, they just kind of brought up 2 points like that are, like here to talk about bats and to just introduce bats to this audience. The first point is like when you were talking about in mice, all these studies in mice, is that one of the issues, one of the key differences between mice and humans is that the mice, they have this really short lifespan, right? Like there's small animals, and in mammals, smaller animals have shorter lifespan, but bats are actually an exception to that. So small bats can live long lives. So there have been bats that have been found that are the size of a mouse. They weigh even less than a mouse, like 10 grams, which is a very, very small bat, that have been found to live for 40 years.
40?
Right? 40 years, 40.
40 years.
Yes,
oh
yes,
my
exactly.
Gosh.
So they have this extended lifespan. So we're hoping, like through a large consortium that we're part of called Bat1K, we're actually saying, well, the genomes of bats are going to help us find out the secret of mammalian youth, right? Of the ability for bats to, despite being really small. So in mammals, larger animals, elephants live longer lives, whales live longer lives, but bats also, for their size, they live longer lives. And if you ever need to advocate for bats, you just need to tell your audience that if there were no bats, there would be no tequila.
That's right, the agave flower, right?
And also that if you kill bats, you're going to kill the answer to the eternal youth. So that's what we want.
And tequila. So we can't do without it.
Everyone has priorities.
Everyone has different priorities.
What's the point of life if you no longer have youth and tequila?
Exactly.
Oh my goodness. Angelique, we spoke almost a decade ago now. It's amazing that so much time has passed. And at the time, you had published a paper on multiple sclerosis, and you were interested in the immune system. And I think you were doing some work on the immune systems of preserved mummies at the.
Correct.
I think. Yeah. How did you go from multiple sclerosis and autoimmune stuff and mummies to bat? I mean, bats are in caves too. Is that like the mummy bat connection? No. How did this happen?
No, I just kept the Halloween theme going. So the answer is right here. Liliana, who's my spouse, is actually a bat specialist. She's a chiropterologist. But my interest in the immune system makes me one of these rare people to whom you can throw an alphabet soup of genes at, and I will know what those genes are. And so I can kind of get the full picture of where, what gene does what in which pathway fairly quickly. And that's actually why we combine forces when the pandemic hit. We wrote with Deanna and our colleague, Dr. David Ray at Texas Tech. We wrote one of the rapid National Science Foundation grants, which are part of the CARES Act for COVID research. And that's where our study of the immune system of bat came into play.
That's wonderful. So we've got Liliana, you're the bat expert. We've got Angelique, your immune system and genetics. And Deanna, how did you come to work with the two of them and work on this subject?
Yeah, I have been interested in the immune system in mammals. I am interested in host pathogen interactions and evolution within host and evolution from the pathogens. And then I started to get interested in bats because, well, they are like the perfect model to study host pathogen interactions. And Liliana and David, I was lucky enough that they got this grant to study longevity in bats. and that it's also another thing that I'm interested in. And I entered to this amazing group. I'm studying. My skills are in bioinformatics, genomics, and immunity.
So if I can jump in here, this is also. So one of the things that we were talking about all these viruses that are being found in bats, and I became really just kind of curious: are those viruses at all part of an immune system within a bat, or is it just that their immune system isn't affected negatively by it? And I bring that up because in infant children, for instance, before an immune system has really gotten a chance to take root, there's viruses there that are doing the job of an immune system.
Go for it. Go. No.
I guess I think I'm gonna start, and I'm sure Diana and Angelique have a lot to say about this. It's not as if I think the evolution or the interaction between the bats and certain viruses is a very long-standing one that has happened over millions and millions of years of evolution. And because these bats, when to me, it keeps getting back to this life history, right? Because bats are not this kind of, born, mature, reproduce, and drop dead. They're not. They kind of survive, right? Like there's this evolutionary advantage. There's this adaptive advantage to a bat being able to survive another round of reproduction, another round of reproduction, another round of reproduction, and reach these long lives. And that's really very different from the models that we normally use as humans, which is mice, which is very different, right? Like these animals just kind of are born and then they get to reproduction and then they immediately start declining, right? So when you think about it that way, there's an evolutionary advantage for when bat species in the wild come into contact with certain viruses, right? To evolve, right? There's an advantage to surviving for the next round. So to adapting your immune system to these challenges. And that is one of the things that our research that is about to be published in Molecular Evolution, that's our starting point in looking at what's special about that immune system of bats that is very different from our own. There is some parallel to what you're saying about children in the way that immune system operates. And I just want to kind of seg into the people who really understand the immune system better than I.
Well, so it's not that the immune system is not there. It's just that it's different. It's not primed. So when you have an immune system that's not primed by a series of pathogen, it kind of is ubiquitous. So it sort of has these mechanisms where the cells are a lot more active and a lot less discriminatory in terms of the pathogens. So basically, so like they will go for anything that that sort of is foreign to the body of the host. But what makes the bat super special is actually something that children also have, and that as we grow older, we start losing, is that they have less inflammation. And as we grow older, of course, we get more inflammation, right? And this is why any kind of diseases that are novel are going to affect us far worse as an older adult than as a young child. Not automatically, because, of course, there's always that clever pathogen that's found a way around that's adapted to that. And in fact, we're actually seeing that with SARS-CoV-2. It seems now that it's affecting younger people more often. And that's because it's adapting to the fact that older people are no longer they're starting to get vaccinated in high concentration. So it's like this is harder to get to survive, and it's adapting to that very situation. So Diana can tell you more about the inflammation in bats.
Yeah, I'd love to hear more.
We have found that actually it seems that the key of success in bats are that they are able to regulate the immune response. So it's not going crazy. sometimes the immune response is the one that it affects more than the pathogen. Sometimes immune response is that aggressive that it can come to chronic inflammation, tissue damage, tissue death, and eventually
they have stuff like the cytokine storm.
cytokine storm.
Yeah.
Right, where it's like that, it just goes crazy, and then your body is really disadvantaged because of that. But
and it's your immune system, system doing that, like in a runaway chain reaction. Yeah.
And the bats don't do that.
They have inflammation, but they don't have like a crazy inflammation like us or like other mammals. They regulate this inflammation because they have lost many genes that are involved in the activation of the crazy inflammatory response. Now the new question that we have is, okay, so now which genes are the one involved in the inflammation in bats that are making like less aggressive? And it seems that all sorry.
Oh no, sorry. I was gonna say, is that because they're the only mammal that flies?
Oh, that's a great question.
Oh,
because they're like
right into there.
most mammals, we run around for a little bit and then we just stop. But they have those wings that they're constantly moving. So if they have I get sore just going for a brief jog now.
Yeah, actually, that was going to be my next comment, that this seems to be related with flight, because flying is a high metabolic cost. So they generate a lot of reactive oxygen species that in us can cause cell damage or DNA damage, but bats have evolved really good mechanisms to repair all this cell damage. So one interesting thing is that it seems that this amazing immunity response in bats, or this amazing regulation of the immune response, is actually like flying can ended up favoring this regulation. Because can you imagine if you are the inflammatory response is activated either by pathogens or allergens that you were talking about, allergies and at the beginning of the show, toxins, but also it can be activated by endogenous signals caused by cell damage or DNA damage. So can you imagine bats are flying and they are generating all these reactive oxygen species? Can you imagine if they are responding every time to all of these bioproducts every time they fly? It will be impossible. So it's like co-adaptation in this flying and immunity. It's like everything's connected.
Is there I had also seen that there were within the paper some questions related to diet and like what bats are eating and how that has affected their ability to defend themselves with their immune system.
So one of the things, there were kind of two big findings of the paper. Like the first is sort of to confirm and corroborate with a much bigger sample, something that we had already that had been reported previously, and then last year we reported in another paper, and it's that panel C that you're showing on the screen, and it's that these pro-inflammatory genes that are found in other mammals. They're conserved throughout mammals. We have them and mice have them, and they are completely missing from all the bats that we examine, right? Mm-hmm. So this had been done before with like 6 different species and a couple of other species before, and thanks to all of our work, and especially Diana's, we actually confirmed that with 37 different species of bats that we went there, and they were of very high quality, and we confirmed that. And so that's for genes that were really missing in bats, which is interesting because it's a way of evolving, or a way of being different, in which what the way that you're evolving is you have missing pieces, right? Mm-hmm. And it was kind of like our the paper has a lot of it is devoted to the interpretation of what would these missing pieces do, what predictions we can make that can be tested down the line. But the second part of the paper was actually about genes that are present, but they either expanded or contracted, meaning the genes are present in bats. It's not completely missing. They're there, but they seem to have kind of expanded. Gene families expand when, whatever it is that those proteins are doing is maintained, right? So it has some kind of selective advantage, or the opposite. And those, we were it was very interesting that for the most part, those weren't immune at all. So whereas for the missing genes that are completely missing in the bats, we got a lot of missing genes that have to do with the immune system. When we got to gene families that are expanded or contracted, what we saw instead is that they were metabolic, or primarily metabolic. So they were taking us in a different direction. Instead of showing us variability at the immune level, I mean, we did find one or two that we might get into the details of that were immune, but most of them had something to do with metabolism. And of course, bats, even though ancestrally all bats seem to have been insectivorous, and most bat species in today's world are insectivorous, there are a few lineages of bats that have switched their diet in a dramatic way. And one of them is the agave specialist, right? These are bats that feed on nectar exclusively, or they feed on fruits exclusively. And that's a huge demand. When you think about a bat that is feeding on nectar exclusively, just consider it's kind of like a human that would switch their diet to just soda. Just imagine if you like switch your diet to just kind of like...
That doesn't work for people. I think people have tried that. I mean, we had one of those big giant 2-liter sodas.
Yeah. I think we've tried that and that doesn't work. So that doesn't work for us because we're not evolving and we're not flying. We're not being energetically active to the level that bats are. But for bats, so we proposed, and that's what we found. We found a couple of genes that are presumably, that have to do with this handling of the glucose, right? And the handling of glucose that seem to be like gene families that seem to be duplicated in groups of bats that have independently evolved the ability to feed up mostly on the sugars, this kind of simple sugars that come out of fruits and nectar. So that's another avenue. But the difference with the first type of results is that these results are within bats. So different species of bats corresponding to different adaptations to their diet. Yeah.
And did there seem to be any, basically all of these bats had lost the immunity genes that you're talking about, just to kind of across the board?
That have to do with inflammation, all of them. Yeah, all of them.
Yeah.
So there was no variability there. There was some, the genes that we found, that the immune system genes that we found that had some variability in them, meaning expanding and contracting, were really, really special ones. And I'm going to let Diana talk about those. But I can talk about APOBEC if you want, Diana.
Yeah, well, one of the ones that we found was the MHC class 1. That the MHC is a major histocompatibility complex, and it's involved in recognition against viruses or intracellular pathogens. So these genes were expanded in some bat species. That is something also that I think is really important and really amazing about this order, because not because some bats have adapted to their own virus, it means that all the bats are adapted to that viruses. Actually, there are some viruses that are lethal for bats, it's like the Takari virus or some Lisa viruses, and actually some filovirus has been related with a mortality event of bats in Spain. So yeah, bats are not like immortal against viruses. That's something
so as much as we now want to think they are, they're not. They are. Yes. They can be affected by some species.
Yeah, and they are adapted to their own virus. If you put a bat that haven't been in touch with another virus, never in their history of their evolution, is going to be susceptible. There's a high chance that it's going to be susceptible for that virus. So that's another interesting thing about this, that the immune system, it seems to be species specific because the interactions are species specific, but these losses are constituted for all the order. That's something, and the MHC is one of that are expanded, and we think our hypothesis is that it might be expanded in order to overcome this lack of inflammatory risk. Well, this dampen in inflammation. Yeah. Because it's going to make more efficient to recognize viruses. And there have also been studies that we have found an insertion of 5 amino acids and 3 amino acids on the MHC class 1 exclusive from bats. And this insertion could help bats to recognize longer peptides for viruses.
Okay, so with the expansion of this major histocompatibility complex, they're able to recognize, and so their bodies, if they come in contact with something, they can potentially recognize it and then do whatever immune response needs to happen. It's just not as big of an immune response as another animal might have.
Or as inflammatory. And the other thing is that these expansions are big enough that they would actually yield a greater diversity of recognition, right? Like the capacity to recognize a wider diversity of pathogens that they may encounter. Then the other immune.
Whereas so like just to pop in for 1 second on the importance of recognition. Whereas what we're dealing with when we talk about a novel coronavirus is that our immune system has no way of recognizing it to fight it off. Our adaptive immune system has never met it, doesn't know what to do with it, and so it's just kind of like, I don't know what to do, and so you're just dealing with the natural immune system, the native immune system.
Even within humans, there are some humans that are able to recognize, and that are the ones that are symptomatic, right? So they seems to be better because they have those adaptations, those variations of the MHC class 1. There was a really cool study about the MHC that they found some variants from the MHC class 1 that are really efficient against this new SARS-CoV-2. So that's important of the MHC. The MHC is really it's high specific. So the more variable you are, or the more polymorphism you have of this family, the wider pathogens you can recognize. So even humans, we carry some humans carries those variables. Some of them.
I want to find out if I'm one.
There is a way. It's just rough.
Yeah, you can still make this happen. And the other group of genes that we found with immune system that had a lot of variability within bats, it's called ApoBec3. Okay. And the great thing about ApoBec, so the apo part is an apolipoprotein, but the ApoBecs in particular, they are a big family of genes that actually specializes in introducing mutations into single-stranded DNA, right? So they are specialized. So ApoBec3 in particular has been found in model systems, has been found to cause this mutagenesis in response to viruses. So you can imagine like there's a single-stranded DNA that's coming, it's coming from the back translation of an RNA strand that's coming from a virus, right? And it's floating around. And then there is this molecule ApoBec3, or several different ApoBec3s in the cell that's capable of introducing mutations into it and rendering it less effective as a pathogen, right? So in bats, what we found was that some bats seem to have a lot of different ApoBec3s, like 13 different ApoBec3s. Some bats have fewer, right? Like so primates, including us, we have 7 different ones, right? But then some other like dogs have like 3, right? And in bats, we see lots of variation from having like 3 to having like 13. Lots of expansion in these ApoBec3s. And we hypothesized that this variation is associated with, just different adaptations to dealing with viruses in their natural environment and to coping with those viruses by using this part of the innate immune system, right? That's just responding by introducing mutations into pieces of DNA that are sitting around. Yeah, and how it forces viral adaptation. And so one of the consequences of this that we're exploring now, or we're hypothesizing and we're in some follow-up research, is that this coevolution. So you can imagine you're a pathogen, right? Like you're a virus. Like your job, you're just like a piece you're just a piece of genetic information with a little bit of a capsid around you. That's it. That's all you are. You're just trying to get your genetic information into a cell so that they make more copies of you. So that there would be this evolutionary arms race that you would enter between the bat ApoBec3s and the virus genome, right? Such that the genome, because the ApoBecs have a recognition pattern, right? There's a particular sequence of this DNA that they recognize, right? So that the genome would evolve away from that recognition pattern, right? And therefore, when this particular virus finds itself in a different host, when it accidentally, when it lands in a different host, it's going to have a pattern of evolution that is shaped by that evolutionary history with the bats, with the ApoBec3. So we're exploring that idea Of course, it's right now that's nothing more than a hypothesis, but it's an intriguing one because it takes us back to the evolution of this ApoBec3 family.
Yeah, and that kind of gets back to the study that Justin brought up right before your interview from the Swiss researchers and the bats finding MERS in that population of bats.
MERS related.
MERS related.
Yes.
In that population of bats. It just, there are bats, like I said, there are bats all around the world. There are massive bat caves in Arizona, and we talk about Southeast Asia. And so would it be necessarily then, say, drier ecosystems, wetter ecosystems, how those environments tend to, I guess, tend to affect mutations because of adaptations as the bats are evolving in these locations, and then how various viruses come along with them along in these different locations. And so would we expect different populations around the world to have different apobecs?
Yes. So what we found. So we're not thinking about environment when we're talking about host environment, not necessarily the climate conditions, although climate conditions are important because, of course, the entire planet, we know that for mammals, there's a lot more species when it's hot and wet, right? And a lot fewer species when it's cold. And there are no bats in the North Pole, and there are no bats in Antarctica. Right, so we do know that there's a very strong.
There aren't parkas that are small enough for them.
It doesn't work like that. So we do know that there are this diversity. These are called latitudinal diversity gradients, right? And bats follow the latitudinal diversity gradient. So there's, to the extent that that's true, that's the case. But what we're seeing is that these ApoBec3s, right? They're very just like the MHC1 that Diana talked about. The ApoBec3s are species specific, meaning some species have a lot of them, some species have fewer. And what we think is happening is that this is going to shape the evolution of the viruses that coexist with those species that have the particular apobecs. And it's going to shape like what even the genome of those viruses looks like, the sequence that it has, and therefore the potential for when it finds itself in a different host environment, meaning in a different species of mammal, to evolve rather rapidly, because it's got, because it's no longer facing its longtime adversary, the ApoBec3 of the bat.
Right.
It's now facing the ApoBec3 of the camel, or the ApoBec3 of the civet, and it's not quite the same game that they had with it. It's not the same arms race that they had. This is very, very hypothetical at the moment, but we do the basis for thinking this way is that variability that we find.
So what does this mean for, how we as humans go about thinking about these populations of bats as we're searching for new coronaviruses, possible reservoirs of other diseases that could have pandemic potential?
And if I could add on to that too, we're looking for these reservoirs. I don't even know what the range of a bat is. I have this comic book picture. They live in a cave. They come out. They go somewhere, and then later they come back. I don't know how far they went. So how big is the region of their territory?
Yeah.
And
well, some bats went as far as Hawaii, so that tells you the range.
Some bats, the hoary bat from North America twice colonized the island of Hawaii, and it's the only endemic land mammal of Hawaii. And the genetics tells us that it didn't arrive there the once and reproduce. There's two different groups of hoary bats that at different times colonized the island of Hawaii. So some bats have very wide range, like the hoary bat has this super wide range that includes the island of Hawaii. And some bats have very narrow ranges and are just distributed in a relatively small area, or they're tied to a certain resource. So for example, the ones that pollinate the agave nectar, they actually migrate along the flowering of the agave nectar, like up and down from Mexico into Arizona and then back, right, following the flowering of this resource. So they're very
They're
tight.
like the monarch butterflies of the bat world.
They're like,
Absolutely.
is that I guess that's migratory then.
Yeah,
They have
those are migratory. Yeah, but there's other bats, but not all bats like that. You know, like it really varies. So, for example, there's an endemic bat, a flying fox that's endemic to the island of the Comoros, and it's there in the Comoros, and that's the only population that there is of that species, livingstones. So it really varies. So one of the things to sort of take in, that's, I think it's very difficult for most of the public to contemplate, is that when we're saying there's 1,400 species of bats, is there 1,400 species of bats? They're all different. They're as different. In many cases, you know, we're talking about being as different as a dog is to a fox, is to, you know, like a wolf. Like it's, you know, some are more closely related, some are more distantly related, but they're just, you know, like envision, for example, like imagine if you have a jaguar and a puma and a tabby cat pet, and now multiply that, that's 3. Now multiply that times 500. That's how many species of bats there are, and they're each unique, just like you want to say, oh, here's that cheetah and here's that puma, they're the same thing, they have the same viruses. You would never say that. You would never, ever say that. And when we say bats, that's what we're saying, right? Because we're not, I mean, there are certain commonalities, like what we found in our report, which is that there's this missing genes that have to do with inflammation, right? But the individual species of bats are very divergent, right? So if you know, like imagine all those big cats and small cats that are different species, and now multiply them until you reach 1,400, and that's how many bats there are. Bat species are.
And so with this study, there were 37 bat genomes that you looked at. I mean, how representative of the 1,400 species do you think this 37 is? It's like scratching the surface of what's potentially there.
Yeah, I mean, we tried our best. So if you look at this picture, I mean, I you know, we're, you know, like we can just give us more money, we'll keep studying that. Okay. but if you look at this picture, we have a blue and a red bit, right? And those are 2 big groups of bats, right? One group of bat, I mean, you don't really need to know the names, but there's 2 big groups of bats.
The yin and the yang.
The yin and the yang, I know. This man called Carl Koopman helped name them. So one is called yin Pterochiropteran, the yin bats, and then there's yang Pterochiropterans, the yang bats, the yin and the yang. And so this blue and the red, they're 2 very large groups. One has the flying foxes and the horseshoe bats, and the Cressionectaris. How do you say what's the common name of Cressionectaris? Anyway, some tiny bat that's amazing, right? And then the other group is it's bats around the world that we have heard about. It includes like the little brown bat, the big brown bat, the vampire bat, the tequila bat. Yeah, the Desmodus rotundus, the tequila bat. And they're all in this other group called Gymnochiropteri. So we tried to, within these 2 large groups, as many species as possible that were as divergent as possible. But yeah, we have a long way to go before we take in the great diversity.
And this is just a shop question. What's the platform that you're sequencing on?
Oh, that's a really good question. So, Diana, you want to talk about that?
Yes, go.
Yeah, it's for this study we use PacBio, Illumina, but the Bat1K use a lot of more platforms. They combine the Hi-C, Illumina, PacBio, BioNano.
So we use optical, just to kind of translate that into English. So we have short read technology, like high accuracy short read technology, high accuracy long read technology, and optical mapping. And that's a Bat1K standard. However, we're like the scrappy American team, and so we went with the short read and the long read, and we're now working on the optical mapping for some of our future things, but we didn't get it for this one, for the new genome that we're reporting in this paper.
And so were these genomes part of Bat1K already, or did you add to Bat1K in the process of doing the study? Or
we use some genomes from the Bat1K and some from the Broad Institute.
Okay. Yeah.
And
sorry, go ahead.
Oh, I just want to say we generate 1 genome for this study. We are taking advantage of the public data.
Mm-hmm. Yeah.
It's already available. That's
Yeah,
great.
we took advantage, and we also had to account for the differences in quality, right? In the quality, I mean, there's a lot of variability. That's why the BAT1K, the 6 genomes that BAT1K that we published from BAT1K last year are sort of a gold standard, right? And we were trying to demonstrate that we could using just the short read and the long read, we could get to like 96% of the gold standard, right? Like we wouldn't get to 100%, but we could get close enough. And now we actually have several more genomes in the pipeline, including all the technologies. Yeah.
Because that technology has been evolving so rapidly in the last 5 years even, that if the public database is mostly 15 years old or something like that, the quality is not going to be anywhere near what you can get in a nice afternoon on a nano or overnight in an Illumina.
Yeah, ours, we actually had nanopore in there as well, because we wanted to see how nanopore. Yeah, exactly. We wanted to see how the nanopore performed in this. But the prices of, I mean, you probably covered this. You know, PacBio was bought out by Illumina, and PacBio prices have like come down as well. And PacBio is higher accuracy than the nanopore, and we see that. You know, we see that in the results. Yeah. But it's amazing. I mean, we really, so BAT1K, our next big thing is the 21 families. So we want to have every single family represented. There are some families of bats like Crestionicteris, whose English name escapes me right now.
These bumblebee bats?
The bumblebee bat. Yes,
That's
the
right.
bumblebee bat. Like the bumblebee bat, which is like a single species in a single genus, in a single family. Or like, for example, Rhinopoma, which is a family of bats that's found exclusively in deserts in the old world. And so we think that there's going to be adaptations that are like really unique adaptations to this water balance being very different. And there's no way that we could explore any of that because we simply don't have the genomes of the species yet. Yep.
As the costs come down, I just can't wait to see more and more and more genetic data. But, you know, then this is Diana, this is your future in bioinformatics. Data management and analysis.
Yeah, no, we have too much data. We need more scientists.
Yeah.
Data is too much.
Yeah. It's all the data. So to go back to the question, though, all this research into the bat immune system and what you're learning of which genes are no longer part of their genome, no longer acting as part of the inflammatory part of the system, not regulating that. How do we use this information? What is this kind of information tell us, or how does it inform us in our future pandemic avoidance?
Well, so the point of this paper, or the way we can take this paper and move it to a translational domain, which is basically take this and maybe use the information from the paper to translate it to humans, is we could actually find treatments, for example, to prevent that famous cytokine storm, right? And the treatment would be block the genes that are missing in the bats, that are allowing the bats to be tolerant, block them in humans so that humans become more like bats and they are able to avoid the cytokine storm. And it's really important because you know, a lot of the antiviral therapies that were pre-existent to COVID have proven to actually really fail. Think of remdesivir, for example. A lot of noise was made about remdesivir early on, and it's like, oh, it's gonna save. Like, it failed for Ebola and it failed for SARS-CoV-2, which is kind of sad. And there's a reason for that. Is that instead of targeting the cell mechanism of entry of the virus, etc., which can be extremely deleterious to humans. Imagine blocking the ACE2 receptors for humans. wouldn't go so well.
No.
And so instead of doing that, be more like a bat. Downplay the information, and this has actually been shown when patients were put on dexamethasone, which is a dampener of the inflammatory response. They were doing much better than on any antiviral that is currently available. So for that particular kind of viruses, respiratory viruses, which lead to a cytokine storm, the way to go is to make people more like bats by dampening their inflammation.
So it's either dampen our inflammation, or we can go the guided evolution pathway and make us all flying, nocturnal, vegan creatures. And then maybe those adaptations will come.
Omnivorous,
Exactly.
though. We gotta be omnivorous. We gotta go after we have to also drink blood, eat nectar, and insects.
Those are all different
Not
bats,
all
though.
at once.
Those are all
You only choose one.
Are they all specialists, or are there some that do kind of?
There are, especially, I mean, the group that I sort of started my career and have continued my career is called the neotropical leaf nose bats. That includes the agave, a bunch of nectar bats, and it also includes the vampire bat, in addition to a bunch of insectivorous bats. And in those bats, there's a lot of omnivorous bats, meaning bats that can eat insects and can mix and match. They can eat pollen, they can eat fruit, they can eat nectar, right? There's a whole bunch of very successful, very wide-ranging bats that are omnivorous, but not with blood, though. Yeah. Yeah, the blood specialists, the vampire bats are specialists. They're completely specialized.
They're specialists.
And I was about to ask if they live longer than other bats, but I'll leave that one.
Well, they actually kind of do. I mean, their body size, the common vampire bat, the Desmodus rotunda. Yeah, the common vampire bat does have a longish lifespan given its body size, right? We measure that in something called the longevity quotient.
Mm-hmm.
And it
But
tells
it's
us how much
interesting
of them.
because the vampire, the Desmodus, the common vampire live longer, but the other vampire life doesn't show a higher longevity quotient. So again, it's species-specific adaptations.
So if we all are doing the vampire diet, I mean not the vampire diet, if we all do the bat diet though, it's going to be tough to do the bat diet because we'd have to. There could be anything we almost choose.
Yeah,
pick one.
Mm-hmm.
Yeah.
Yeah, there's a frog-eating bat too. Again, among that group of bats that is the neotropical leaf-nosed bats, it includes the frog-eating bats, for instance.
That is your favorite, the carnivorous bat.
There's all these carnivorous bats actually that are specialized. It includes the fishy. A close relative is a fish-eating bat as well. So the family includes a huge diversity, and then its close relatives also have some diversity in their diet. Mm-hmm. Yeah. So
it's amazing. I mean, imagine with 1,400 bats to choose from, you probably have examples of just about every dietary style somewhere within there.
Yeah. We have exclusively nectar. We have nectar with some other things. Exclusively insects. Exclusively gleaning insects. Exclusively like on-the-wing insects. All kinds of specializations. Fascinating.
Yeah, because now I also want to know everything about bat microbiomes.
Oh yeah, absolutely.
How those are related, because that's an incredible
and their metabolism too, right? I mean, let's not forget that the immune system is ramped up or ramped down by the metabolism, and so that the diet has something to play into.
Yeah, so they fly. Do they have songbirds especially have a higher body temperature, and that is part of how their immune system works, is just by having a naturally higher temperature. Do bats have a relatively higher temperature? Is that part of their metabolism?
Yeah, I mean, Diana, you want to talk about the flightless fever, hypothesis?
Yeah, it can go to 47 Celsius, the temperature from the bats. flying.
Whoa!
So it can emulate the fever in us.
Yeah.
And that's another hypothesis that why
Whoa,
they
that's
can regulate.
116 degrees.
Yeah, no, that's egg cooking time.
That's not just a fever in us. That's dead.
Yeah. You boil over.
Yeah, so there's this hypothesis that Diana should explain that's called the flight as fever hypothesis. And
That bacteria and pathogens cannot survive to that temperature. I mean, not even us. So that bats that maintain these high temperatures, they avoid this viral replication or bacterial infection. But that's a hypothesis.
Yeah, that's one hypothesis. But the corollary of that hypothesis is that if a virus has evolved to live in bats, to survive these high temperatures that some bats can have, then when that virus finds itself in another host that is not a bat, that doesn't reach that temperature, then that other host's fever is not going to do anything to it.
Nothing.
Because in the bats, it was a normal thing to every day at the peak of activity experience this high temperature. So that's a flight as fever hypothesis.
Now I want to go searching in those high temperature bats for extremophiles.
Yes,
Yes. Exactly. Yeah.
That and I'm with global warming and raising temperatures too, the general environment that bats find themselves in will have higher heat. That'll make that maybe more possible to encounter in the future as things evolve to handle. Probably not viruses. Maybe it's more just the general pathogens or bacteria and the like. That'll like slowly be able to handle higher and higher heat as the temperatures
and again,
rise.
I just want to emphasize that bats are just so. There are so many different species. So, for example, one of where I started my research was actually in the islands of the Caribbean. And I don't know if you've ever been to Puerto Rico or the Dominican Republic. The islands of the Caribbean. There's this very special ecosystem that the bats have created themselves, and it's called a hot cave. Have you heard of hot caves? And in these hot caves, so
Have we talked about this?
So imagine a karst. So imagine like karst, and then water runs at the bottom, and it kind of opens up a cave, but it's closed it forms a cul-de-sac, right? And then bats colonize it in the hundreds and thousands, and they colonize the bat cave, and they raise the temperature inside the cave, and there's usually running water at the bottom, so the humidity reaches excess of 90%, right? And the temperature is extremely stable. So whereas, we're just discussing how bats, for example, can, their temperature can raise, there are some species of bats, for example, in the Caribbean, that they actually must live in that hot cave, right? They must, like, when the hot cave disappears, when somebody pierces a hole at the top or just erosion pierces a hole at the top and the cave disappears, they have to go find another hot cave to live in because they require this very high humidity, very high temperature environment to maintain their metabolic, to maintain homeostasis. Mm-hmm. They must live inside these environments. So again, we have to be kind of like really cognizant of this variation. At the other extreme, we've got bats that are adapted right up here in New York. So one of the things that one of my students is studying is, of course, white nose syndrome, right? Mm-hmm. These bats are hibernating, and that's kind of the opposite, right? Like they live in these caves and they drop their temperature and they change that. So we must be, this is like a call for understanding that bat diversity, because without understanding that bat diversity in the ways that they live, in the ways that they adapt, we're not going to understand how this virus is circulating.
Right. Yeah. And understanding how they move within individuals within populations, how they move between populations, how those populations come into contact with other species.
Exactly.
That all of those interactions.
Right. That's exactly right. So we can't just say, well, climate change is going to do, raising the temperature is going to do this. Well, we have to understand what the bats are actually doing.
There's so many pieces to the puzzle that it's not an easy.
But if you go to the Caribbean, look for the hot caves. Ask around. People know.
That would be amazing.
But don't go in the hot cave.
No, please don't go. Don't go in the hot cave.
No,
do not
That
go in the
sounds
hot
like
cave.
hot death.
No, don't go. Yes,
it is.
Go in with a gas mask, maybe.
Well, we actually, yes.
You have to have a respirator.
You have to have a respirator.
and just know that you'll be wading in poop up to your waist.
Yeah, it's like guano. Like guano. This hot cave. I mean, some.
We have the best vacations.
Yeah, we have the best.
It sounds like you have the most amazing vacation.
There's some hot caves. Okay, there's like hot caves at the past that you can like drill down into the guano into the last, 10,000 years of the history of that bat poop, because that's an environment. Again, it's a very stable environment in which the bats are going in there, and this is the environment that they must live in. And not all bats. So just to bring up, not all bats live in caves. No, lots of bats don't live in caves at all.
They roost in trees.
They roost in trees. They live in other environment, or they live under a bridge, right? So there's this flexibility, right? Like,
The most important thing is don't encroach on the bat's territory. That's let's stop having human encroaching into the bat's territory, and that's the most important part.
Well,
and let the bat be, because they're rendering a tremendous service to humanity. Without bats, there would be no humans left, because the food source will disappear. The pests will overtake, and the more we encroach on their territory, the more problems there's going to be.
I think that's a wonderful note to kind of start winding this up on. We're going rather late, and I know you're up late right now, so we're just having so much fun talking to you. So just for each of you, if there's just a final thought about bats, or about this paper in particular, and what you'd like people to understand, what you want people to take home with them from this conversation. Diana, do you want to?
I think I will say that stop blaming bats, stop being afraid of bats. I mean, it's normal to be afraid of the unknown. Just embrace them and learn about them, and respect bats.
Do not embrace them too close, though.
Embrace them metaphorically.
From afar.
Metaphorically. It's a metaphoric embrace.
Embrace by donating money for genome.
I love the dual message there. Yes, you need to get to know bats, embrace them, but stay away from one. Don't be scared of them. But if you see one, don't go anywhere near it.
Yeah, it's like there's these messages. I think we should use that meme that Diana made. The meme that Diana made from her, that really is the meme from her Twitter talk. It's a really good one that's the message of the paper. I don't know if you can project it.
I think it is. Hold on one second.
If you can't project it, that's really.
Is it this one?
Yeah.
No.
Yes. Yes, that's
It's really they're just bats are different. Their immune system differs. It differs from our models that we've studied. So we've cured all these diseases in mice. Mice is our model for everything, and it's also different from ours. So it's a different, but it's amazing we get to share this earth with these life forms that are completely distinctive, and that have this combination of traits that no other group of mammals has. And so there's that diversity there that we have only begun to explore. There's so much more to learn from bats. Yeah.
And to finish this, did you know, Kiki, that our first date was actually having a field trip in the Caribbean, a hot cave?
No.
And our relationship survived.
If you can survive a first date like that, I mean, you're going to survive anything.
Tamana is a hot cave. It is a hot cave. That's right. Tamana Cave in Trinidad is a hot cave.
I think it was a trial by fire.
The secret of love is getting buried in guano.
Yes, exactly.
Guano.
Yes, guano happens.
Oh, thank you all so much. It has just been wonderful to get to speak with you, and all of the information that you've shared. And I do hope that people do take your words with them and appreciate bats and stop being so afraid and start appreciating them for their diversity and for all of the things they can offer. Like, really, they could be our saving us from the next pandemic. They could also help us live longer.
Yes.
Yep.
Yes, that's definitely because
it's all upsides.
And if you're thinking of a field of study that will allow you to choose any destination in the world, apparently, some really nice spots, study bats.
Yes.
Yes.
Don't end up just studying that thing that only lives in a desert. You'll get tired of deserts eventually. Don't only study the thing that's way under the water. You're not going to get into its environment very often. Study bats. You can travel the world, and there's always something to study, it sounds like.
Absolutely.
Especially 10,000 years of poop in a hot cave.
Well, that's year 2. You don't bring that up to the first year. That comes later.
That comes later. All right. We will post your Twitter accounts and a link to the paper on our website. And I just want to say thank you again. It's just been really, really wonderful getting to talk to you tonight.
Thank you for having us. This was brilliant.
Brilliant.
This was wonderful.
It was
Thank
really
you.
fun.
Have a good night.
Bye.
Take care.
Bye.
Good night.
Good night. All right, everyone. This is This Week in Science. We are moving on to some more science stories. We have more science. Hey, do you like this show? You enjoy This Week in Science? Yeah. Well, maybe tell a friend and help share it with a friend today. Share TWIS. We can all enjoy it. What's Justin doing? I hope he's getting a story, because it's his turn to talk about science.
Were we supposed to bring more stories? I felt like we already brought the amount of stories. Oh!
I thought you had one story. Come on.
So, I do. I gotta find it. That's why I'm stalling. Oh, yeah. So this is underwater archaeology. University of Texas at Arlington is part of a research team that's studying a 9,000-year-old stone tool artifact, or a bunch of artifacts, I guess, that they discovered in Lake Huron that originated from an obsidian quarry that was 2,000 miles away in central Oregon. So the obsidian flakes from the underwater archaeological site, they think, is the oldest and farthest east confirmed specimens of Western obsidian ever found in the continental United States. It's just one of these things. First of all, you had to go, again, to the bottom of a lake to find it. But yeah, 4,000 kilometers away is another way to say this. Or, of course, 4,000 kilometers, for those not familiar with that, is 4 million? No, 400,000. Oh, God,
So-
now I'm messing. 4 million.
So wait, I'm confused. So this,
kilometers.
the quarry was in Central Oregon, and the
Yeah.
lake was
Lake
2,000
Huron.
miles away.
2,000 miles away.
How'd it get there?
This is,
What's going on?
It floated, apparently, or just got moved. I don't know. They're not explaining it. Very small pieces that have very large stories to sell. So it could be that this was trade routes and the rest of it. It could be. I think that's actually what they're going to say. Yeah. This site was anyway, the point is the site was underwater. That was the whole point of bringing up that story in the first place. We need more underwater archaeology, since water levels are rising and have risen over history. So much of our history as humanity is now below water, being such a shore-bound peoples close to the sea. I don't know. I gotta go sneeze like 4 more times. I'll be right back.
All right. Justin's allergies are giving him some problems. So let's have a calm moment and talk about birdsong, because birds are amazing. Researchers are using zebra finches to try and use brain signals to translate birdsong into, birds' brain activity into song. Birds sing songs. Their brains have vocal centers that have been shown to be pretty much in parallel to human vocal centers. There are all sorts of things that are very similar. And so the neural signals that are involved in creating a bird song, from its initiation in the bird brain all the way down through the motor neurons to the syrinx of the bird and the song that actually is produced, there are parallel structures, very similar, in the human brain that allow for the production of our speech. And so to be able to someday be able to interpret brain activity and produce voice sounds for people who have paralysis of the vocal cords or paralysis from neck down that does prevent them, or locked-in syndrome, or any number of disorders that keep people from being able to speak, that potentially we could take neural signals and technologically produce the sounds. So these researchers publishing in Current Biology, they are at University of California, San Diego, and whoa, my computer just freaked out. I hope everything's good. We still there, everybody? Woohoo, I'm still here.
I see you.
You still there? Okay, good.
My
computer just turned off. My screen just turned off for a second. I don't know what just happened there. Anyway, the researchers used representations of the bird's vocalization patterns, and they are mathematical equations that model the changes in pressure and tension in the syrinx. So they're using these algorithms to map the neural activity to the representations. So it's this kind of mirroring of the neural activity to actually turn it into something that could be song. But in the end, they were able to create zebrafinch song based on their work, and they're going to try and model it more closely to be able to create better vocalizations and actually get more nuance. But the simple representation allows the system to learn mappings that they say are more robust and more generalizable to a wider range of conditions and behaviors, so that they're hoping to be able to take this new technology and not just create bird songs, not just record bird brains and then play bird songs in your speakers. No, but to help people who need to speak in a number of different situations, which will be pretty cool. And then my final story has to do with vitamin D, because it's summertime, and at least here in the Northern Hemisphere, and vitamin D has been a topic of interest for quite some time when it comes to research into health. A new study in Science Advances in mice, I'm going to say that right now, study in mice, not in people, found that mice with low vitamin D levels had exaggerated cravings for opioids, and they felt the effects of the drugs more strongly. And so the researchers publishing in Science Advances, they first looked at data from about 20,000 participants in the National Health and Nutrition Examination. It was a survey. They looked at these retrospective data sets. And then a second study had 3,000 patients that were admitted to a hospital between 2014 and 2016 that had an opioid use disorder, and then 8,000 matched controls. And so they looked to see whether or not people with insufficient vitamin D were more likely to have opioid use disorders. And they were. So they found that in the first data set, patients with insufficient vitamin D were 50% more likely to use opioid painkillers compared to patients with normal vitamin D levels. Those that were deficient or severely lacking were 90% more likely to take opioids. And those that were in the data set for the opioid use disorder were more likely to be classified as vitamin D deficient or insufficient, which I find very interesting. So they thought, hey, we should look at this further. And this is where the mice come in. They used vitamin D deficient mice that had a diet deficient in vitamin D. They also had transgenic mice that lacked a vitamin D receptor. And then they looked at these mice and how they wanted to how much they wanted to use morphine as they passed through a morphine chamber during a conditioned place preference test. And those that had the low vitamin D spent more time, or those that had the low vitamin D and no vitamin D receptor spent more time in the morphine chamber than other mice. They took this even further, and they wanted to know how the mice responded to the opioids. They put these control and deficient mice on hot plates to measure how their pain response worked. And then they gave them morphine and ran the test again. The vitamin D deficient mice stayed on the plate longer, and then the morphine acted more effectively as a painkiller. So their pain response was different, and the morphine also worked more than mice that did not have the vitamin D. And the effect was reversed when they restored the vitamin D levels. There were more experiments that they did. They linked it to sunlight and UV radiation as well. And the bottom line to all of this is that this is potentially knowing somebody's vitamin D status before coming in or when they come into the hospital. You might know better how a patient will respond to opioid painkillers, how much more likely they might be to need more of them than somebody else. And so vitamin D could potentially be a cheap and easy intervention moving forward. And something that could be, it's not something that would need a lot of clinical trials for doctors to be able to start implementing it also. But this is really just a fascinating link between, the UV, the way that vitamin D is produced in the body as a response to UV exposure, the vitamin D receptors, and then also kind of this linking of when you're in the sun and your body's making vitamin D, there is an endorphin release and you feel better. And so there's a link there also with the opioids. If you don't have the vitamin D, opioids can also make you feel better. And so there's a really interesting link in here between sunlight, vitamin D, and opioids, which was really curious to me. Very curious. Take your vitamin D, folks. Yeah. If you're in the summertime season, wear your sunscreen, because that's good. But, spend some time. Make sure you've got your vitamin D. I know I will. Justin, you look like you are done. Like your allergies, you need some vitamin D. You need to go to sleep and then get some vitamin D tomorrow.
So I'm fine. I can't see anymore because my eyes are watering and my nose keeps running, and occasionally I have to sneeze. Yeah, I don't know what it is, but something in the air is bad for me today. I need to go consult with the bats. Like, I need a little less of a reaction.
You need a little less inflammation is what you need.
Yeah, a little bit less would be fine.
I think you do. I think you need a little bit less of a reactive immune system at this point in time. Yeah, maybe not just helping us in our immune and our cytokine storm responses. Maybe the bats can help us with are just normal allergies, because those are getting worse. Oh, everyone, we have made it to the end of another show. And I do want to say thank you for listening. I hope you enjoyed the show tonight. And many thanks to our guests tonight, Dr. Angelique Corthals, Liliana Davalos, and Diana Moreno Santillán. It was just a wonderful conversation, and I hope that you all learned a lot. Shout outs! Time for shout outs. Shout outs to Fada for your help with social media and show notes. Gord, thank you for manning the chat room. Identity 4, thank you for recording the show. Rachel, thank you for your assistance. And I would love to thank our Patreon sponsors for all of their generous support to the show. Thank you to Pierre Velazarbe, Ralph E. Figueroa, John Ratnaswamy, Kira, Carl Kornfeld, Melanie Stegman, De Kramsta, Karen Tozzi, Woody MS, Andrew Bissett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Lee, Ellie Coffin, Maddie Perrin, Gaurav Sharma, Shubru, Don Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Shauna Nina Lamb, John McKee, Greg Riley, Bart Cassenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Mark Mazzaros, Artyom, Greg Briggs, John Atwood. This profile name is hilarious in the context of some other podcast. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Sutter, Philip Shane, Kurt Larson, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And for those of you who are interested in supporting us on Patreon, you can find that Patreon button at TWIS.org. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, broadcasting from our YouTube and Facebook channels, as well as twis.org/live.
Live! Want to listen to us as a podcast?
Sure.
Just search for This Week in Science wherever podcasts are found. And if you enjoyed the show, get your friends to subscribe too.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org, or you can also sign up for a newsletter.
That might come out quarterly.
Eventually.
Yeah. You can contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twisminion@gmail.com, or Blair at blairbaz@twis.org. Just make sure you put TWIS in the subject line so your email doesn't get, lost from the immune system of a bat.
You can also hit us up on the Twitter, where we are at TWIScience, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
And we hope you'll be back here again next week for more great science news.
And if you've learned anything from this show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list
list of items I want to address. From stopping global hunger to dredging Loch Ness, I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science, This Week in Science, This Week in Science, 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 This Week in Science, This Week in Science, This Week in Science, 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 This Week in 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 Science.
And that is the end of the show. We are in the after show. Justin has disappeared. You stayed and watched the end of the show.
We
Look at a bird. That's fantastic.
Oh, Justin's back.
Justin's back.
Justin's back again. Yes.
So this is not on the air?
This is on the air.
We are
but
still on the
we are
air. This
still
is
on
called
the
the
air.
after show. So in the after show, we sometimes will ramble on about the story that we didn't cover, the point we didn't make that we should have, or we look to the audience who's in chat to see if they have questions that they didn't get answered, or if they have more inquiry, that sort of thing.
Oh, I see.
Oh, I see that. Yes,
Yeah, there's like a...
From broadcasting from the Bat Cave.
Yes.
Yes, we this should really be our background, but yeah.
The Batcave
it's-
background. You should have a Batcave background.
Yeah.
Right? I mean, why didn't we think of that?
Next time.
So, but this actually isn't. This is not a background. I am actually broadcasting from the moon. I don't know if we mentioned that.
Yes. Lives on the moon.
That's awesome. And the vacuum is not at all affecting you.
No,
The vacuum,
no,
not an issue.
apparently I'm allergic to moon dust, though. An oversight.
Aw. Yeah,
Yeah,
that's
when did that happen?
This has been like an ongoing thing where I laugh at people for having allergies because I'm not affected by anything, and then suddenly I was.
yes. Well, yeah. Allergies are cumulative, right? So you don't have it until you have it.
And I've done nothing to avoid them my entire life. So yeah. So I think what happened was it cooled off a little bit last night. So I just opened all the windows. And I'm like, this is nice breeze and everything. So there's something out there that's getting me.
There's that one species of plant that's killing you.
And I just gotta wait till that plant or the mold or whatever the thing is gets knocked down again or finishes doing what it needs to do, and then I'll be fine. And then I'll go back to making fun of people with allergies. But until then, it's a serious problem. You should not make fun of it. It's really horrible.
It's awful, and then
And I took all that because I don't even know what I went and got, like the super strength. This will knock out anything that affects you're not allergic to, and it's either not kicked in or it just doesn't seem to work on. It's the wrong stuff. I don't want to say the brand, but it's a major brand of a thing that you would use to drill down on an allergy.
Ah, yeah. Well, take 5.
take more.
Is that really what it is? You just like if it's not working just because I used to know people who used
a
to.
day and a half, but that's okay.
There
you
was
know
one,
what?
wasn't there one brand that you had, it wasn't like it wouldn't work in one day. You were supposed to take it for like a week or two before and it would build up and
Maybe. Yeah.
slowly build up in your system to block your allergy response.
So I'm just doing it wrong. I'm just an amateur at this, is the problem.
But what you can do right before bed, if you've got the little pink Benadryl pills, those will knock you right out.
There it is. Yeah, or as Kiki mentioned before, be more like a bat.
Be more like
a bat.
And that's the thing. I'm not eating my local honey. I used to do that a lot. Maybe it's been the honey the whole time. Now I have to do a whole experiment where I hope this pollen stays there while I try different honeys and see.
for your N of 1.
Yes.
Yeah.
But you know what? Oddly, that's all I care about in this experiment. That's the only problem I'm trying to solve is the N of 1.
Yeah.
Oh my goodness. Good night, Fada. What was that?
Good night. The mice thing is really interesting that you got that in the beginning of the show, because every time I present on the multiple sclerosis research, which has evolved now, and I'm actually partnering with a pharmaceutical to put that into translation.
Yeah.
And,
interesting.
But every time I present on it, my one slide is why mice models don't tell us anything. And so I have all the explanation as to, if you're going to be talking about metabolism, you can't use mice because mice don't have the same metabolism as humans. To start off with, they're missing an entire component of the metabolism of lipids. They don't have the HDL component. So why do people keep using mice when they do metabolic?
Because they have short lifespan.
Yes, exactly.
Because you can get them,
and they're easy to breed.
So you also order them from a catalog to.
That's not a good reason.
Well, it's not.
It's a convenience.
The convenience.
Instead of bat, I'm going to tell you right now, it's a lot more convenient than a bat. If I had a catalog that I could pick from and said, send me this, it costs money obviously, but send me this 20 bats, right? And they're all kind of similar genetically, or they've been sort of bred through many generations to be more or less the same, and I can standardize them. That's like a dream.
Yeah, I was really fascinated. We interviewed some folks from the Jackson Labs. It was as I recall, a pretty intense conversation about just what they're capable of and on what scales they're operating.
Yeah, and you can knock in and knock out genes. Well, everything, but there's so much that you can do. It's a wonderful model. Why do we study the model? Well, it's like you can use them anyway, whatever.
I am dreaming of the day where we don't have to use those poor goddamn mice and rats, and we actually able to have imagine if you could have a panel with all the pathways, and just put your compound and see it goes through all the pathways like a pinball machine, and see what it knocks and what it doesn't knock. That is my dream.
So it's just basically
silicone.
With no organism, but just it's just the molecular genetic interactions. Yeah.
Well, yes. At some point, you're gonna have to test it.
Yes, eventually, but
yeah.
At least you'll have a better, more robust in silico testing capacity. So if there's any here in the ether,
in silico,
yes.
That's what the supercomputers are for, right? To be able to
so
test all the different combinations and see what things bounce into other things.
The machine learning wants to do it. The only problem is we have to give it that information. We have to give it every parameter.
Yeah.
In the first place. And if we were having this conversation, gosh, I don't know if we've been doing this show, so if it was 10 or 15 years ago, whenever, when we very first started the show, I don't even think I think we were just maybe beginning to get into what the microbiome affects. I remember stories microbiome has a connection to your brain. Wow, and now it's like a given how much that interconnection is actually responsible for, oh gosh, even behaviors or desires to seek out nutrients or regulating everything. It's such an integral part. And then everything that we've learned about nutrition, though, all those books can't even be opened now because it's not straight molecular. There's this whole other environment it has to pass through to have an effect, to interact, to. So yeah, we're still at the beginning of so many of these things.
My microbiome only wants cake.
You have the cake microbiome. That's good. Mine is chocolate. Give me all the chocolate. Yes.
Well, being Belgian.
That's true.
What would be a one step better than a mouse that's currently, or what's the ideal still animal model? Right. I guess it depends on what we look at, right? Because there's hamsters that are good for liver?
Golden hamsters.
Yeah, the hamsters are better. Actually, it depends. Like hamsters are better for a variety of things. Infectious disease, particularly for vaccines, hamsters are better. But of course, if you really want a much better model, then you go into.
Then you're in primate, right?
Yeah, primates or pigs actually are pretty good, but very uncomfortable to move into those models. And of course,
It's a big burden.
It's a huge burden, so you don't want to do that. That's why the mice are also the easy.
Yeah, I was working. I had a high school job very long time ago, and I was working in an orthopedics research lab, but they had all sorts of other cross collaboration stuff going on. And I remember the lab director telling me about working with, especially the larger animals, because they were doing. One of the things that they were working on is actually became the robotics behind how they do hip replacements now. But they were taking volunteer dogs, people's pets, largely, as well as kennel animals or animals that were going to be put down, that had hip problems naturally. Like I think they were working mostly on labs. They have this is like a thing with
shepherds,
all labs.
Maybe.
They get this.
Shepherds, yeah.
Yeah.
And so they were testing us out, and it was early, and not all the dogs make it, and that sort of thing. But one of the things he was telling me about was that his hope was for just a database. Mm-hmm. Just a database of all the research that was performed on animals, and it's morphed into my head as a null result database, where you can see all the things that didn't work.
Right.
So that if you've got an idea of something or a pathway or an experiment that requires you to use an animal model, you could easily access and see if it had been done before. And maybe it's been done multiple times. Maybe it's been maybe this idea has occurred to people 50 times, and they've run this animal experiment in 50 silos of research. Never worked, so they never published. Mm-hmm. Yeah. But knowing that could prevent so much unnecessary time, money, and then of course the need for those animal models as well.
Yeah, that's actually brilliant. I think that's something that I find problematic, is that failure is never reported. Mm-hmm. And so you keep trying to reinvent the wheel of that failure because it hasn't been reported.
It hasn't been talked about. Yeah. So it would just be like a searchable. The problem is who would host it and pay for it. For all of them, it always comes down to this money thing.
the failure
results, right? The journal of results, where you can.
The journal of null results, and just to have a searchable database. If you're thinking about your experiment, you plug it in, and then you see, okay, wow, this is already. And then how much does that speed up if they're well done? That's the other thing. Is you assume it's well done research, or see a number of times it's been done. And see the methodologies that they use, and say, okay, now I'm looking at this experiment that didn't work. Here's something about the way they performed this experiment that I would have totally done differently, and I only need to tweak 2 or 3 or 4 factors, whatever it is, and now maybe it's worth running again. Or, wow, this is a really well done experiment. It had the null result. Now you can shift focus to another approach without having to have gone all the way down that path. I think it's brilliant. I think it would accelerate research. The problem is nobody wants to pay for it. No one is wants to.
And no, and it, this would actually also make the life of reviewers at granting foundations or granting agencies, federal agencies, so much easier.
Yeah.
Because they could literally go throughout the different experiments and say, aha, this is a possible research, and not found 5 or 6 researchers that may be doing, like, repeating an experiment that is being shown.
That isn't going to work. Yeah.
And then on top of that too, you could also cite why you didn't do something.
Right.
Yes.
With a null result, and then so it's worth publishing. You may get you may get thousands of sightings of your paper's failed experiment because everybody looked at that and go, and here's why we didn't look at this. And now you're cited for your research. It had nothing to do with your research. Isn't all about the result anyway. It's you doing the work. The work is still then recognized.
From the perspective of, the publisher parish environment of academia, where would it count to publish a null result, like in the Journal of Null Results? Like, I know there is
Eventually, I think it would.
There is a journal that does take like failed experiments. I wonder if it would actually count to be able to do that.
It would actually, because
It would. I think
it would also increase, like, if you're getting hundreds of citations that says, hey, this person didn't, failed, and this is why we didn't do this.
Yeah.
It would increase your H index, right? And
Yep.
universities look at the H index for tenure, so it could help. But if you have colleagues, so the tenure thing just starts at the department, and what your colleagues generally do is they look at the title of the journal, not your H index.
It's true that as it climbs, as the information for tenure cases climbs up, becomes more and more quantitative. There's an old saying that deans can't read, but they can count. Right? So, like, your colleagues, they can read. So they can actually kind of read, like, is this a journal that we think is a good journal, or is this not? But as it travels up the chain, then it becomes a much more numbers game. So I know.
It becomes a numbers game, and then also my understanding is it also
numbers game.
Also needs to be published in English for it to.
Oh yeah.
Yeah.
This country, I know.
Yeah.
I know all my publications in Russian didn't make it.
They didn't make it. But it's an interesting thing. There's I've seen a new trend recently, at least in ecology and evolutionary biology, to people kind of probing that and quantifying, like, how much people who are not speaking English, how much they're being held back. So I'm originally Colombian, and there was a Colombian researcher who published, like, in PLOS ONE. I think in PLOS ONE she published this thing about how many barriers as a Spanish speaker, how many barriers that Colombian scientists, I don't remember how she did it, but anyway, she did this analysis and put it in a scientific paper, how much. And then imagine, that's like, what if you're Indonesian, right? Like, Spanish and English share a lot. There's a lot of similarities at some level, right? But what if your language is just completely different? The barriers, I imagine, have got to be much greater, right? And so people are only, at least that I've become aware of, only very recently started to probe that, how much that's impeding science, right? Like the fact that everyone has to be adopting this lingua franca, right? But it's not equally easy to everyone to adopt it. So there's this barriers.
There's that, but also the algorithm that does ranking will put a Spanish published paper with more citations, with more Well, yeah, basically with higher levels of citations, lower than English ones with fewer citations that are less recognized. I
wouldn't find that surprising. I don't really know how it works. I've seen that sometimes when I search for something, it tells me like exclude all the things that aren't in English. And then I think to myself, no, I'm like exploring. I'm exploring the history of Latin America. I actually need you to give me everything that you can find, not just the English language.
Yeah.
For example, give an example. So yeah, I wouldn't be surprised if this was one of the byproducts of the way these things are optimized.
Conversely, there's also the big deal right now, since we're running out of antibiotics, right? The big deal is returning to the phage approach of dealing with infection, where you train your phages for very specific bacteria, and that actually is research that scientists in Russia, the old USSR, had published in the 1950s.
Wow.
It took people reinventing the wheel now in the 2000s
Because they couldn't read.
Because they couldn't read the Russian research to redo that whole research.
Yeah, I mean, it's kind of, English has become, because I think, Western powers it probably has to do with Western powers and dominance and colonialism and all sorts of influences, but yeah, English has become the common language as opposed to any other, and it's unfortunate, but I mean, from a perspective of just doing this show right now, it's wonderful that everybody was able to speak the same language to be able to have this conversation. And it's like
absolutely.
We could have tried French, and I would have stumbled through it with great difficulty. But
so would I. I cannot. I mean, all my scientific life has been in English, and so when I have to explain to my mother, for example, what I do for a living, which she's constantly disappointed because I keep stumbling on words, and I don't know how to translate these words.
She probably thinks you're inarticulate.
Yes, I know. She definitely she's the kind of person who would tell me, the reason why you're not finding a job, in parentheses, I had a job. You're not finding a job is because you're both inarticulate and you can't spell right.
In French.
In French.
Yes.
Thank you, mother. This is
okay.
Nice. Yeah, I mean, it's it's sort of like the mother's high standards. It doesn't matter, that you have a PhD and that it's from a fairly good university. It's like, there's like massive deficit. Yeah, there's massive deficiencies in your.
But it's interesting because sometimes I wonder, like with this business of the languages, like I actually had, I could have totally missed that story for that book that I co-edited.
Oh, yeah.
The book about, so I have this kind of parallel scientific career where I studied deforestation in Latin America. And a few years back, and particularly in the Amazon, and then a few years back, I was actually trying to get, the Andean countries, there's an arc of deforestation that happens and that it's associated, or a lot of people want to make the argument that, coca cultivation, coca is the plant from which cocaine is made, right? Mm-hmm. There's only 3 countries in the world that produce this plant for this global legal market, and they're Andean countries. And so a lot of people wanted to make the argument that, you could stop deforestation if you could just stop the planting of this plant, this coca cultivation, if you could stop it like that. Mm-hmm. So I wanted to kind of get behind that, like try to understand that better, go back from that. And I started kind of probing into some early history, like what happened before the cocaine boom, right? Like what happened. And 80% of all the documentation was in German.
Oh my gosh.
Yes, like 80% of the papers, these were geography papers that were like some that were from Switzerland, some were from Austria, some were from Germany. Everything was in German. There have been all this sort of a big bump, like in the 1960s, starting in the late 50s through the 70s. There had been this kind of spurt of research, like German language dissertations from around the German-speaking world. So you can imagine, like, graduate students from Europe are like landing in Bolivia, they're landing in Peru, they're like living in Putumayo in Colombia, and they're moving there and documenting these projects to move the population from the Andes down into the Amazon. And they're like doing all of this, and maybe there's like one tiny abstract in English, and everything else is in German. And
you do a search.
Yeah, and I don't really remember. I think I came across, like, I was searching, like I was doing this very old-fashioned thing where instead of just Googling, I was actually kind of searching in the library, and I came across something, and I thought, okay, this is like inaccessible. I'm gonna have to interlibrary loan it. And I loaned like 20 different things, and they start arriving, and they start like all in. They have this amazing maps, and they have this detail, like, for example, like the gender disparity, like it was mostly men bringing into, in some places there would be like 75% of the people who moved in were men, and like this, and the economy, like what people were producing, this very, very detailed social science. And it's all in German language. And there's only like a tiny, like less than 1 page abstract. And I thought, wow, I could have missed, like I could imagine like I was going to find a lot of things in Spanish language, right? But there was a lot in German. And it was
missed this amazing.
I could have missed this amazing story. We actually ended up editing a book with a colleague of mine who's an historian. We published this book that was about that history from different perspectives. Mine was like an environmental perspective. Other people had, and I could have totally missed it if I had limited myself to English language documents or Spanish language documents. So it was just like a series of fortunate accidents that led me there. And so now I know that there's this gigantic history of Latin America
in German.
That's in German language. If you want to find out what was happening in the outskirts of Santa Cruz in Bolivia in 1965, you better shape up
on German.
On German language. Like I think it was a fascinating thing to code and to document. Yeah, I was not expecting it.
No, yeah. I mean, I would have expected it just, it would be local languages, people, Spanish language, and the idea that all of the researchers who would have been doing the documenting would be from Germany. That would be, that would not be expected. I wonder how many things like that, because of people going different places and who's doing the documenting and who has the time, the money, the ability to actually make those notes in different places. How much has been missed because you go, oh, foreign language paper, and you throw it away. Yep. I'm just not going to include that.
Come up. Yeah, or it's just not.
Or it doesn't come up in the search at all.
The search is not giving you that, right? Like it was for me. It was more like like you when you grab that thread. Like I grabbed that thread. Like somebody in English is citing that work, and I'm looking at it, and I'm like, oh, it's in German, but I'm gonna try to see. Like I really want to see what they are saying. And then from that, and scrolling and just doing this kind of very old-fashioned scholarship, right? Like very old-fashioned kind of like, it makes me feel ancient, like what we used to do before we did web searches. I don't even remember, but this world that existed, right?
I know.
So it's like following those leads until I got to this.
Library cards.
So it was amazing. I'm glad I did this history, and it's been really helpful. It's been enormously hopeful to sort of bring that scholarship into the front. And I'm wondering now, when Angelique was at the example of the phage in Russian, right? Like how much we're missing, like when we're not searching. There's a giant scholarship, for example, in Brazil, right?
Yeah.
A lot of journals that are published in Portuguese. Still, that's large scholarship.
I think if we went back to the journals, the Russian journals of be surprised about how much there might be there for cancer, for infectious disease in general, because they were actually fairly advanced.
Yeah.
Yeah.
They were doing a lot of really interesting advanced work.
Right, and they had
Yeah.
they had a huge budget. I mean, they had literally had unlimited budget
Yeah.
for research, and it was all behind the Iron Curtain, so nobody ever saw it. Yeah.
Well, there it is. There it is. Search in other languages. Let's look for things in other languages.
Don't be afraid.
Don't be afraid. Don't be afraid to.
Like, translate all of the Russian journals from the 50s and 60s and 70s. And,
I mean, it's just such an impediment, right? Like, it's all about, or like collected, it's just something Justin said about sort of doing the things that people have already done, right? Like, if you just kind of access, right? Like,
Yeah.
access the minds of these people that the stuff is already written, the experiments are already done, and we just don't.
Yes, and it is getting better. You know, there's a lot that still needs to be unsiloed. But it is getting better. But I have a number of times tried to. I was working for a research company, and I was trying to come up with they were making the ask for people to present new project ideas, right? A new product and new project things, venues to research. So I went delving. I'd get an idea, and I'd go and I'd find, oh, we already did that. oh wait, I've got another idea. Oh yeah, there's something else that does that better. Okay, and it was it was very defeating, but I never came up with the idea that didn't work that way.
right.
I might not have ended up with a good idea, but I didn't end up with presenting a thing that the company, another company, had already done, tried, put a patent wall around, or whatever whatever the outcome was. It is extremely important. The Google Translate stuff is getting better.
Yes.
As well. But yeah, if that stuff, if those Russian documents, if that German text is still, bound within a book somewhere, it might. Yeah, then that does sound like a neat project.
Google has not yet taken all of the books into the internet. So.
And the journal. So I relied for that kind of research. I ended up relying a lot on interlibrary loans, where somebody physically, took their stack, and scanned it and sent it to me, and then I had to do OCR. Like if I wanted to use Google Translate, I had to. So it was like several layers, like OCR from a scan that somebody did, and Google Translate. But mostly, I just had Angelique help. With the real critical stuff, I just kind of got the gist of it from this crazy Google Translate, and then I was like, does this really say what I think it does?
Yeah.
It is tough, and I don't know if the technology for scanning is quite sophisticated enough, or if we even have. Some people may not remember this. There was a point in time when we tried to do what the internet is now doing in preserving text. It was called microfiche. And I don't know what we did with it.
They show it in movies. In movies, people go to the microfiche, like movies that are like, oh, it's 1970, and the research.
Don't know what we did with all the microfiche. I'm afraid we put it all in an under air conditioned warehouse somewhere, and then one hot day it just all fused.
I don't know. I have hopes that all the microfiche from the libraries is all kept in a climate-controlled storage area. Where you can, yes,
and it's cataloged well.
It's cataloged well. Exactly. I mean, all the funding we give to our libraries.
You know how difficult it is. Do you know, for example, for genomics, how difficult it is to just keep up with data? Which is the data that's generated, and keep it organized and keep it. It is so difficult.
Yeah.
It is so difficult. And it's not, I'm not even talking about the stuff that's like in hard drives that fail. I'm talking about like the format changes. It is a battle, right?
Wow.
Yeah.
Yeah, but I mean,
That's actually the problem of librarians. So
Yes.
I was completely baffled when I was looking at the library at the Museum of Natural History when I used to work there. I said, well, what's your biggest challenge? Is it the air conditioning? Is it the space? Is it the stacks? Is it the blah? And I said, no, it's none of that. Our biggest challenge is obsolete technology. And like obsolete computer technology, so obsolete formats, obsolete, I mean, we all know that we started with the floppy disk and we still find the floppy disk drive, and then,
Yeah.
at some point they say, okay, the CD-ROM is going to be the gold standard for archive.
Yeah.
and if you're thinking that the cloud is doing all those things, I have news for you. Because, for example, we actually send like DNA out to a company that sequences it, right?
Yep.
And they charge us because they buy a hard drive and they ship us the hard drive.
Mm-hmm.
Just because it's easier than loading it to the cloud.
Because it's just
yeah.
faster. It's faster for
Wow.
them. Yes, I'm not even. I mean, there's a lot of companies that are also making use of the cloud, and they kind of like.
Yeah.
But at some point, the quantity of data is such that they just put a line item. They're overwhelmed. They just put a line item, right, and just put it on a
Here's
drive.
your hard drive.
They ship us a hard drive, is what they do.
Yeah, that seems like the best possible solution. Yeah.
Yeah, think about that.
Mhm.
Like this age of the information technology and like this flows, it is easier for some companies to send us a physical hard drive that could fail or whatever and could lose all our data than for them to upload it to the web and to wait for us to download it and then release it.
Mm-hmm.
It's changing, but it's just we can barely keep up actually with the growth, the data growth, right? Like we have difficulties keeping up. We're constantly having to upgrade for storage and to actually it's like in our field of science, which is not very translational and that used to be very kind of natural history oriented, right? It was relatively hard to find just pockets of money where you say, I just need to constantly upgrade. Like I need to constantly get more computer equipment.
But
Yep.
citizens do too. I mean,
Yes, true.
when you're thinking about the space that a photo used to take 10 years ago, and now,
Right.
With the pictures being like 25 pixels, megapixels, you go from taking 0.5 megabytes to almost a full gigabyte for a specific photo. And that's the same thing.
We have the same situation.
Upgrade your hardware to keep up with the technology that you're using.
Yeah, I mean,
Know it.
they do.
And they want you to buy the new gadget.
They do, but I mean, that's also, keeping up with the data is one thing, and being able to keep up with, you're collecting new data, and like you mentioned, you get new formats and things have to be reformatted. But then, and Justin mentioned, genomes from, 10 years ago, say 15, you're 10 years ago, you've got these genomes. How are we dealing with the preservation of all this data? Like, how do you make sure that it is preserved for perpetuity? And that's got to be just a huge, it's not like just printing something on acid-free paper.
Yeah, well, for the genomes, the government is doing a pretty good job with NCBI. I mean, I don't know how they've managed to scale it up, but NCBI,
But they are.
The genome archive has grown. It's really a pain to submit to some of these repositories is really a pain, but the fact that they still exist and they haven't collapsed, that's already to me like props, mad props to them for that.
And that they're still nimble enough to actually fit in with your own data searches.
Yeah.
That
So
is amazing.
The functionality of NCBI has just been growing and growing and growing, and it's like it's got three-dimensional models, it's got all these awesome things. And then there's this short read archive, which is a misnomer because it's including PacBio. There's like giant long reads in the short read archive, and that's also growing explosively right now. And so that's the federal government right there. That's federal government infrastructure that is holding all this information. Yeah, so
That's great.
Publication level information, right? It's more the pre-publication level. The pre-publication is kind of like when I get those reads and they're in my lab, and I need to make sure that the people of the future have access to maybe the stuff that doesn't end up getting published.
Yep. Exactly.
Even the repository that we use for publication, like Dryad, which is basically where you put your raw data in, so that
like tables, supplements,
and literally like the raw data, so that other scientists can replicate.
Yep.
Even they are starting to like this. They're starting to run out of space, quite literally. Yeah. And they're the gold standard. You cannot publish without putting your data.
You know what decays faster than any of this? Any pipeline that you use, any bioinformatics pipeline that you're using.
It's like buying a car. The minute you buy it, it's obsolete.
God, it's worse. It's like, I don't know. It's like buying a computer, right? It's like you're like like from one year to the next, like the pipeline, like is my environment, my computer environment with all the dynamic library. You're running the same script that you wrote a year ago, and you're trying to run it again, and you get dynamic library 22 error. It is so. I mean, there are tools, like there's SnakeMake. There are tools. There are tools out there to try to grapple with this. This is the thing that decays the fastest.
So, and this is the other thing too. You can't be just a natural scientist and work with this stuff because you need somebody in there programming R or whatever, or working with your Bytes. But I was watching the, I don't know if it's where I call it the IT team working with the bioinformaticians, and there was like 4 different programs they were fine tuning just to put the reads that they were getting into a reliable format. And that just, absolutely mind-blowing how much time, space, and effort and energy that part of the job takes up and away from the science.
There's how there's some concerns, or there's been like a debate in the popular literature of like, is artificial intelligence going to take over the world? And,
right.
Like kind of displace humans or something. And every time I think, so this when we were presenting that phylogeny, right, that evolutionary tree of the bats with the 2 groups, right? Right. Okay. So that's been my bread and butter as a natural scientist for a long time, is that I know how to make phylogenies. All right, so let me explain to you that the taxonomy of species changes over time. And so something has a name, like the genus and species name, but the same genus, the genus may change over time, or people make typos. So for example, if you have a data point that comes from a geographic location, and you've got a sequence that somebody published, and you just want to kind of pull it all together, right? And they have typos. And I would say that back in the day, when we were making when I was making phylogenies, kind of compiling the data, not with the big pipelines, but I was compiling by hand, about 40% of my time would be going into putting a single name. And to this day, like when I put that in a, when my students are trying to automate these things, it's like that's the step that requires manual curation.
That still requires manual curation.
There's a manual curation step that's unavoidable. The sort of homologizing taxonomy and dealing with the typos. There's like this manual curation step, and I'm thinking every time I see that, I'm like, the artificial intelligence is not going to take over anything. The computer can't recognize that this is a typo, that, this 7-letter word is matched everywhere except for this one letter. Like, I can't, no, it doesn't work, no. It requires a human being, right?
So, and isn't this is also, well, partly a problem with that 10, 15-year-old data that's in there, where people couldn't wait to name a gene something.
Yeah.
But their coverage compared to...
Or the names of genes. Oh my God, the names of genes. Oh my God, the names of genes. That's like a water.
That's a.
Yeah.
Oh God.
Oh, your reaction is priceless.
No, this is... So, my significant other is a bioinformatician that runs across this with mostly with working with fungi, but yeah, all the time, where somebody somewhere named a thing at some point, because it's the first time they'd kind of gotten it sequenced, maybe, or fifth time or sixth time, whatever. And so even like the names of what genes are and what they're purported to do, it's almost like it's terrifying. It's terrifying because as much data as we have, it's not necessarily even usable data if everybody...
calling things different things.
Yeah.
How do you correlate if it's...
Well,
yeah.
okay.
Is it Gene Wiki or something like that? Like if you go, like some of these...
Gene cards, but there is also like, so there's a big movement, and actually the National Science Foundation has been sort of funding a series of projects for gene ontologies, right? Okay. And they've also, so gene ontology, so that's an ontology for ontologies more generally, not just for genes, but also for like limbs, like for like organism, right? Like limbs and all of that. And there have been several massive projects attempting to do exactly that, right? Like where you're actually coming into the field and trying to homologize across vast expanses, trying to do that. But
if you look at those things, that every gene looks like a career criminal.
Yeah,
because they have all these aliases.
In this experiment in the rat, it's got this spelling. And over here, there's this other acronym. And by the way,
when it gets to human, it changes name. We actually, when we were doing the data analysis for this paper, the first step was to basically have all the synonyms of the genes correctly. So we various tools online that are now available, but half of the genes that we had were unknown. So we had this block of genes that were unknown to the machine and to the search tool. And yet put it individually into Google, and Google would find a gene card because it's a synonym that has never been reported before. So we had to do that
a lot of stuff by hand.
And you have to be very discriminating, because sometimes there's actually some tools that will, like in the absence, they'll kind of like look for the nearest thing that you can get, and it's not necessarily by spelling, it's because it's trying to kind of like navigate all of these equivalences. And so it would actually kind of give you that a particular gene. Do you remember about 1K paper?
Oh, yeah.
There was that gene. There's that gene that really the function was unknown and there was no real data, but due to some kind of proximity in the database, it kind of gave you all of these answers. Yeah. And you had to kind of watch out. So we would have to fill in both for the fact that the computational tools would miss a whole bunch of genes. But once we got answers some other way, we had to kind of evaluate them as well. And the thing is, with the bats, when we looked at this, there were 700 missing genes. Yeah. There were hundreds of genes that are missing from the genomes of bats.
Wow.
A substantial portion of them are relatively easy to deal with because they're olfactory receptors. And that's something, like this gene family, I don't know how much you know about olfactory receptors. Fascinating gene family. Like this amazing gene family.
Let's have another podcast about
It's
that
another
right now.
podcast. Right now, let's just talk about olfactory receptors, right? The largest protein coding family of genes in mammals.
Right.
It's the largest
Yep.
protein family. So anyway, so that's easy. It's like, okay, there's all these olfactory receptors that are present in other mammals,
But not in the bats.
but are absent from our bat genomes. That's okay. We can live with that. Bye.
Not what we're dealing with
Right,
in
like,
this paper.
Not in this
Goodbye.
paper. Not
Next paper.
bioreceptors. Yeah. Exactly. So that was easy. That took care of a few hundred.
Yeah,
And then we could move on to the
the vomeronasal.
the vomeronasal, this other form of infection that you've heard about, the vomeronasal or Jacob's absorbing, is that when you're walking your dog and your dog kind of goes on another dog's behind, or he goes against the wall like with some pee, right?
Mm-hmm.
Have you ever wondered why the dog is doing that if they have such a great sense of smell?
So they do get rid. I just figured it was
Why do you hold right up close? Like,
yeah.
why do they do that? Well, because
Yeah.
they're using a different sense of smell that we cannot even imagine because we don't have it. It's the vomeronasal.
We do have
Oh, the vomeronasal. Yes, they're getting the pheromones in there. And, yes.
Exactly, and they
Yeah.
and that wet part, right? Like that, it's traveling. It's not
Right by the
traveling.
palate.
It's not traveling as a volatile. It's not traveling in the air. It's actually kind of doing.
It's more physical.
yes.
Right. It's not
heavier
volatile.
particles,
It's heavier.
yeah.
Exactly. It's heavier, and it's traveling like in the moist part of the.
I don't think I've ever heard anyone explain it that way before. That's fascinating.
really tasting the odor. Like,
More
because
tasting.
it's like
Right.
back of the palate, so it really goes through.
So
through
it's a different sense of smell that we don't have. So we don't really have like a frame of reference, but dogs and cats have it. Like lots of animals.
But maybe if we went and we stuck our faces on.
Well,
Let's
not do that. But so the key is that the point is that there's this vomeronasal receptors as well that are also actually, that's a whole other story. Like there's a whole, like bats are interesting because most of them don't have that.
Some don't.
Most of them don't have the so they're like us. Like they've lost, like a whole bunch of primates. We inherited it from our primate ancestors. The fact that we don't have those receptors, they're not active. They're pseudogenized. Right?
But you can still see the place where it was.
Oh, yeah, you can see it.
Still see the vestigial area where the vomeronasal used to be.
Yeah, where is it?
Back where
in the
it's
human,
at-
where is
yeah,
it?
in the human, it's at the back of the palate.
No? Does it have two little dots? Oh, is
That's
it?
so fascinating. All I've ever heard from people, and even going through anatomy courses, and we're searching for the vomeronasal organ in humans. Humans have to have it. There's got to be one, and it's so interesting that there's the place where it used to be. You can see the vestigial location.
That people are searching for it because the channel, the ion channel that communicates the vomeronasal to the brain in humans is broken.
Mm-hmm.
So it's not like it's, conceivably you could have something else take over, right? But that's actually how we studied it in the bats. Is my student stubbornly decided to study the ion channel in the bats, and by studying that ion channel, she discovered that it had been lost 13 times in as many different ways. Like it's just been lost over and over and over and over. Yeah.
Well, I can imagine if I was going to be in a hot cave with 10 feet of...
The last thing you'd like to know is have a very sensitive sense of smell.
Oh, yeah.
It's a pretty
Tamp that down a little bit.
There's a cave in Jamaica that's one of the most biologically diverse in all the Caribbean, and it's called St. Clair Cave, and it's got not call it the Davalos bath. This is an apocryphal ruin. Okay.
It has an area in the cave that has been rebaptized as Davalos Bath. This is where St. Davalos just fell in. That
Oh.
Well, what happens is
yes.
you have to understand that it's like there's water to your thighs, and the surface of the water is covered in guano.
By water, yes.
There's water, and the surface of the water is covered in guano and like dead things that have fallen into the cave.
And things eating the dead
and
things.
and things like critters that are eating the dead things on the surface.
Yeah. It's an ecosystem. There's everybody's enjoying it.
This passage of the cave is called the Inferno Passage because of the temperature and humidity that and how it's a very oppressive heat. And I was just the whole time I was there, I just kept thinking, I was wearing my respirator, I was wearing wet waders, but I was also wearing my glasses, and I just kept thinking like, I might trip over, and then I will be incapable of being any use. I will be useless to this expedition if I travel in my glasses. And I did not bring an extra pair. This is not good. So I kept just moving very cautiously until I tripped. And I was holding a pair of binoculars, trying to kind of like find some bats and stuff like that. So I was holding the binocular. I was trying for the binoculars not to fall into the water, and so I, I hit like full force against the rock, and just forward motion while holding the binoculars, and so I just went in, and then of course the water with the guano just flooded into the waders.
Oh,
the things we do for fun, but we can totally organize your next vacation.
Anyway, it's one of the most biodiverse caves in Jamaica. I think it's a fascinating location, and hot caves are special. They're special places.
Hot caves with guano baths.
Yeah.
I mean, I want to see the vacation posters.
Yes. If you receive an invitation from us, be very wary.
Okay, believe it or not, there was someone at some point who wanted to set up some kind of disco.
Oh yes,
at the entrance of the cave. Somebody,
disco.
Yes.
In fact, one colony of bats in another part of Jamaica was displaced by somebody making it into a disco.
Oh wow.
Now, I don't think it was in the hot part of the...
Keep your discos out of the bat caves, people.
When it's not an area for illicit rendezvous.
Oh yeah, that was the Dominican Republic.
Yes.
Okay.
Where you take your mistress to. I mean, why? There's so many questions there.
Well, okay, yeah.
Quiet. It's secluded.
It's stinky and hot.
And you're gonna come home smelling like bat guano. And not a mistress.
Exactly. Nobody's gonna perceive somebody else's perfume.
That's right.
Oh my gosh.
Oh
my goodness.
Anyway, it's not as bad as Angelique makes it sound like it's really bad. I mean, it's.
I almost died like 5 different times. 5 different times on our first date.
But it's not because of the cave. Well, it wasn't only because of the cave. Okay.
There was a scorpion involved. There
were other things. There was the Ferdelances.
Oh yeah, there's also Ferdelances.
The hot cave is not responsible for the Ferdelances on that mountain.
Danger!
He's going to a judge, and it's already judged that, it almost killed me.
Nothing happened with the Ferdelances.
But it didn't, so we're all glad, because you're...
Yes!
All swell that ends.
All is well.
No.
I think we're getting late here, so what I'm going to do is end our broadcast for everyone. It's even later where you are.
Well, actually, it's gone all the way up to early by now.
It's early.
It's early.
It's tomorrow
That's
now.
right.
Yeah.
So, Justin, say the words. It's good night, Justin.
It's, okay, you're almost there.
I got it. What?
Okay, you got.
Say goodnight, Justin.
Good night, Justin. Say goodnight, Dr. Kiki.
Good night, Dr. Kiki. Good night, everyone. Thank you for joining us for another episode of This Week in Science. We hope you have a wonderful week, and we'll see you next week.